My lab is focused on brain plasticity. Specifically, we want to find out how our genes and our genome controls brain plasticity. As you might know, our brain is not static. In fact, our brain changes all the time in response to experience that we have on a daily level. And it turns out that our genes have an essential role in controlling this brain plasticity. One of the most exciting findings that came out of our research is that we were generally thinking of genes as drivers of brain plasticity. That's what makes our brain change in response to new experiences. That was kind of the general thinking. Now, work that started in the US and that we're now continuing here in lab and that we're very successful with is surprising, exactly says the opposite. That it's actually the genome that kind of tries to compensate for other changes that happen on a different time scale, and that the genome tries to maintain brain function. Now, this is, I find, really striking. One of the classic models where the influence of environment on brain function has been tested is actually in the visual system. Because it's very easy to manipulate visual experience, right? Dark and light. It's very easy for us to measure the responses of the brain circuits to visual stimuli. And so that is the brain area that my lab is focusing on, the visual cortex, testing the hypothesis that the genes that we are interested in do not actually drive change in the visual cortex, but they actually maintain the function of the visual cortex. For example, the way you see the world without these genes would be changing from day to day. And in fact, you can say, well, it's actually important that I see every day that, say, blonde is blonde, or blue is blue, or that a straight line is a straight line. And so what we are now testing is really the genetic underpinning, ensuring that the brain works from day to day in the same way, while still being able to adapt to new experiences, which actually happens. I started my research group here at the Weizmann Institute about 2 years ago, after I did a postdoctoral fellowship at Harvard Medical School in Boston, USA, also in the Department of Neurobiology. We are currently collaborating with the research group at Harvard Medical School, who is actually an expert in these kind of visual circuit measurements. This is a very multidisciplinary research that we're doing in lab. And in fact, the question is, I want to say, nearly age-old, but the ability to investigate these questions over multiple levels is very difficult, and there is actually only few places, I would say, in the world that really have the ability to investigate this question thoroughly over multiple levels of organization. And I think we're very lucky to be here at the Weizmann Institute, where we have the infrastructure and the intellectual infrastructure to really do this research over all levels of organization, and that's what makes the Weizmann Institute a phenomenal place to do this kind of research.